All right, hello everyone. Welcome to the very first Science on Google Plus live conversation. So this is a Hangout on Air hosted by the Science on Google Plus community. My name is Amy Robinson. I'm coming to you live from the Massachusetts Institute of Technology, MIT, in Cambridge, Massachusetts. And I am joined today by some of the wonderful fellow moderators of the community and geneticist Ian Bosdet. So we'll have everyone introduce themselves, and then we'll get started talking about this landmark Supreme Court ruling. So.
Okay, I'll go. Hi, I'm Ian Bosdet, and I'm up here in the University of British Columbia, Canada. And I have a PhD in genetics. I actually worked on Drosophila fruit flies for my PhD. And then I worked at the genome. We have a very good genome sciences center here, one of the better ones in the world, I like to think. And I worked there for, it's been about 12 or 13 years. And right now, in the past, I've worked on sort of aspects of the Human Genome Project and mapping the mouse genome, the ENCODE project we worked on, and a lot of cancer-related genomics. And right now, my interest is mostly cancer biomarker discovery, so lung cancer and hereditary breast cancer mostly. And I'm excited to be here today.
Yeah, a perfect person to discuss this.
So my name is Jason Davison, and I'm the Science on Google Plus moderator for Earth Science. I'm a researcher at the University of Waterloo. I'm working on my PhD on coupled simulations of water exchange between the subsurface, surface, and atmosphere. I'm really excited to start our first hangout for Science on Google Plus.
Hi, I'm Nic Hammond. I'm at the University of Rochester. I'm the moderator for science education on the Science on Google Plus. And my PhD was actually in structural biology, so looking at the actual three-dimensional structures of DNA and RNA. So that was sort of my interest in this kind of thing. Now I'm in science education, obviously, and so how the public sees science is very interesting to me and stuff I look at now. So that's how I sort of got into this.
Wonderful. Excellent. So, and we're very excited also to have everybody who's joining us watching live. So I guess I help moderate the science communication side of the Science on Google Plus community, and I help labs at MIT crowdsource research, primarily with iWire, which is a game to map the brain. So without further ado, let's get started and talk about those wonderful molecules that are in each of the cells in our bodies. So the Supreme Court recently handed out this ruling, unanimously deciding that genes are no longer patentable. A company, Myriad, held a patent for two genes that were associated with breast cancer called BRCA1 and 2. And that patent was overturned. So we decided to put together this hangout to talk with Ian and see what that means, why that's important, why such a ruling, why this got to the Supreme Court in the first case, how it will influence us going forward. So Ian, could you just talk a little bit about why this case is so significant and what it actually means?
Oh, sure. Okay, and I think I said before, maybe I didn't, so I'm not a lawyer.
Yeah.
I'm not an American, so American law is not my forte. So Myriad has had patents on BRCA1 and 2, among other genes, but this case was specifically about BRCA1 and 2. And these are two genes that are known to be associated with hereditary breast cancer. There's a protein model of one of them. And so mutations in these genes, if you carry those, you have a very high risk of developing breast cancer or ovarian cancer. Sometime back in the 90s, there was a big race to discover which genes these were that were associated with these hereditary cases. And there was these discoveries of these genes. And then through a very complicated series of business maneuverings and legal maneuverings and patenting, Myriad eventually ended up with patents on both of these genes. Significantly, I guess they're patents, and there's many of them, but their patents are on the sequences of the genes themselves, not some method to find them or some method to analyze them. But they're actually on the sequences for the genes, and then sequences for cDNAs, and I'll talk a bit about cDNAs in a minute. And for even small segments of these genes. And so they've held these patents for a very long time, and they've used those to be the sole provider of testing, certainly in the U.S. Of testing for these genes. So it's important for patients, if they think that they have hereditary cancer, they can get this test. But Myriad is the only company that's been providing those, and they've been quite aggressive at ensuring that they protect their patents and no one is infringing on those. And then there's been a lot of legal challenges to these patents, saying that you're patenting DNA sequence. Everyone has this DNA sequence, and you can't just patent a sequence that occurred in nature. You didn't really, you found it, but you didn't create it. And so it's been in and out of the courts for years and years, and then it finally made its way to the Supreme Court, who looked at this matter and just gave this ruling in early June, saying that and it was sort of a mixed ruling. There was a lot of things on the table, and they essentially said that Myriad cannot patent the sequence of the genes themselves. They can patent the patents that are covering what are called cDNAs, or complementary DNAs are patentable, and so those are still relevant. And so the scientific community and the legal community and the biotechnology community are all trying to wrap their heads around what this means as far as gene patents in general. There's many other companies, there's many other patents for other genes, and what does this mean for all of those, and what does this mean for everyone going forward?
So you mentioned cDNA, and you actually provided us with a pretty good graphic, so I'm going to share it here. If you could just walk all of our viewers actually what cDNA is, and just walk through the different steps on this infographic that you provided. So let me put this back up again.
Okay, so this is my PowerPoint diagram. Oh, I can't see it now. There. So this is really sort of the central dogma of biology, and I just tried to explain sort of the issues that I think are relevant here. So in box 1 there, I've just sort of shown a gene by that little line at the bottom. And a gene is in the stretch of DNA that makes up your chromosomes. There's segments there's sequences called exons, and in between the exons, the red exons there, you can see these things called introns. And so a gene, you have a human gene or eukaryotic gene, is broken up by these introns, which are not generally considered useful. So it's sort of, I don't want to use the word junk DNA because that's really bad, but it's sort of something that's not useful for the gene's function that breaks these things up.
But if you read an article that says junk DNA, they're talking about that segment.
Sort of, yeah.
Sort of, okay.
It's a really bad term, like you said it, because anyway. So that's a gene, but with those introns in it, it's not specifically useful because the exons are what's important. I tried to make an analogy to a TV show. The show itself is the exons, and the commercials are the introns. So the commercial breaks you want to get rid of. So in box 2. There's this enzyme that converts the DNA into the working copy. So the DNA is sort of like the information in the nucleus of the cell and the chromosomes, and you can't actually do something with it. It's just sort of the store of information. When you want to do something with it, you turn it into an RNA copy. So you can see that little enzyme that kind of goes along the DNA, reads the DNA, and kind of spits out that green RNA molecule. So this is it's very similar to DNA, but it's slightly different. And at the end, you can see you've made this long RNA that has exons and introns in it. As I said, the introns aren't useful, so in box 3, I sort of tried to draw how the introns are removed. And it's a very complicated, I don't understand all of it. I think no one really understands all of it, but there's hundreds of proteins that are involved in splicing out these introns and finding exactly where they are. And they're kind of pinched off and then cut out. And in the end, you get this, what's called a messenger RNA, or a mature RNA, which has none of the introns in it. So that's just all the gene information kind of stuck together and all the bits that are not useful taken out. And then that is something that the cell can use to actually make a protein. So in box 4, just sort of showing there's the RNA again, and then there's this huge structure called the ribosome, which is essentially an enzyme that reads RNA and makes a protein. And you can see the little orange protein coming out, and proteins are sort of the functional things in your cells. So they go around, they are structural, and they're enzymatic, so they make chemical reactions happen, and they do all sort of most of the things in the cell are proteins. and then the RNA isn't kept very long, and so there's a lot of steps to degrade it over a certain amount of time, so that it can be turned over and used. The building blocks can be used again. And then a cDNA, and this came up it's sort of I think the point that they're trying to make in the decision is that, and I've tried to put in there no patent. So up there in box 1, I said you're not allowed to patent. They've decided you can't patent that DNA sequence that's actually in your cells right now. Mm-hmm. And in box 3, you still can't really patent that mRNA because that's naturally occurring. Your cells are making those all the time. Mm-hmm. And then that sort of offset box there in the lab, synthetic cDNA. So cDNA is a complementary DNA, so it's really a copy. It's a complementary. It's not composite. They actually said composite in their original ruling, I think. So for practical reasons, RNA is difficult to work with. It degrades. You have enzymes on your skin and everywhere in the lab that are just going to break down that RNA. It's much easier to work with DNA. We have a lot more tools to do that. So there's an enzyme that's not normally functioning in the human cell that reads RNA, that purple RNA, and actually makes DNA. So usually it's a DNA to RNA, and this is a RNA to DNA enzyme. And so in the lab, you can take your enzyme, add it to your RNA, and you make a copy of the DNA. And so there at the bottom, I've said that cDNA is a DNA copy of that spliced RNA. So this is DNA that's only the exons and doesn't have those intervening introns in it. And so in general, I mean, there's always exceptions, but in general, that's not found in your cells. And this is what the Supreme Court said. A patent is okay on that because this was done in a lab. The technician had to change it or do something so that that molecule can be created. So that's an active invention, and that's patentable. And so I think that's sort of what I wanted to get across with this diagram.
And so how does that impact the state of biotech research and development, that component is theoretically patentable?
Well, I mean, I think it's important that sort of the sequences in the genome and naturally occurring sequences are not patentable. What they were trying to say is that if you modify those sequences, then you should be able to patent that. And unfortunately, I think unfortunately they used the cDNA as an example of a modification. They're saying that, and I think they said that the lab technician removes the introns, and therefore it's modifying. That's an active creation. That's something novel, and so you can patent that. I don't really see that personally. I don't see that distinction because the cell is actually the one that's removing the introns. It happens normally anyway. So really, the technician or it's like format shifting. I mean, I don't want to bring copyright into this, but that's kind of what it. Instead of RNA is DNA. It's a different chemically. It's a slightly different molecule, but it holds the same information as something that's natural. And I don't really see that as really modifying it in a way that should be patentable. And I think that maybe I'm getting ahead of myself, but so the patents have to be non-obvious. I think they have to be novel, and I think one of the terms is they have to be non-obvious to an expert in the field. And I would really say that taking a BRCA1 mRNA and turning it into a cDNA is incredibly obvious to anyone in the field. So I wonder if some of these cDNA patents that are strictly just using that might not pass. They'll pass the hurdle of it's not naturally occurring in some situations, but they might not pass the hurdle of is it obvious.
Okay, that definitely gives a good insight. So we have some questions from the crowd.
Yes, I'll start off with a couple of questions from the crowd. The first one being from Rajin Drani. He asked, does the Supreme Court ruling impact, if at all, any gene patents held outside the U.S.?
I don't know. I think it does in that a lot of countries sort of look to the U.S. and try to follow their lead. But every country or every has their own laws, and as I said, I'm really not an expert on that sort of thing. I think there are a lot of other countries where you can't already, you can't patent a naturally occurring DNA, even if it's isolated, even if it's like taken out of the cell, but it's the exact same sequence as you find. So in some ways, the U.S. is sort of following some of these other jurisdictions in kind of trying to catch up with that. So it probably will. It's going to have effects. I think I don't really know what they'll be though.
Okay. So the next question is from Daniel Martin. Is there any danger that this will reduce investment in gene therapies and gene-based research?
Yeah, I think probably not a whole. I think it might. There's certain business practices which are really just trying to have a patent and then just aggressively defend that. And there's nothing really innovative about what you're doing. You're just sort of first to the post, and because of that, you've got this little monopoly. And I think that some people would criticize Myriad for doing that, is that they were very good at what they did, but they just really had this monopoly, and they weren't maybe innovating as much. So I think that there's going to be some companies that have really gone towards that, and then now this is really going to hinder them, or they've put a lot of work into something assuming that they get patented, and now this is saying that they can't. I think it'll spur more innovative research. Like, you have to change something if you want to patent it. You have to have some novel use for it. So there might be some more motivation to be innovative.
Another thought with this that I guess I was reading from the New York Times article, right, was that to test for BRCA1, it was like $3,000 to test for this one gene that they have patented, whereas you can sequence an entire genome for what, $1,000 to $5,000. And other companies were prohibited from working on that gene because it was patented. And so now other companies will be able to come in and produce more competitive prices, more innovative testing technologies, things like this.
Yeah, for sure. I mean, I think it's probably about $4,000 if you get sort of the full test. And I don't think it's not fair to compare like $1,000 genome to a clinical test. So I mean, that was a lot of money. But some people, I've seen people say, oh, I could sequence that for $100 or $200, and I think that a clinical test that you can make medical decisions on, you want it done properly, and I don't think you're going to see BRCA1 or 2 for $200 anytime soon. Yeah. But it's certainly, the prices have come down. Sort of the day that this ruling came out, there was like 5 or 6 companies, and that they had the press release and they were just waiting to hit send. And as soon as that came out, they just said, hey, we're offering it. And so there's people offering it, like reputable companies that'll do a proper job. They're offering it for sort of $2,000, $2,300. So it's coming down. There's a lot of data to generate, and there's a lot of you have to be very careful when you're doing a clinical test. So some of the price does come in with that as well.
What's the difference between a clinical test and, say, a cheaper one?
Well, I mean,
besides price.
I guess there's a lot of things. I mean, and it's not to say that a test done in a research lab is not careful and is not high quality. Mm-hmm. Clinical testing, there's a lot of procedures to show that your equipment is up to date and sort of accreditation of the facility. You've got proper procedures. You're not swapping samples, which is a big thing. So if you swap 2 samples and that person's now positive and that person's now negative, that has real impact.
So basically, there's quality assurance type things.
A lot of it is that, yeah, and really the analysis as well. And Myriad has spent 20 years analyzing these sequences, and they know a lot about these genes. And that's another aspect that this will open up, is that no one was really testing these genes, and so, or if they were testing them, they weren't being very obvious about it, and so there was no sharing of information. And there's a lot of mutations out there, and a lot of times you'll sequence one of these genes and you'll find a mutation you don't really know what it means. It looks, maybe that's bad, maybe it's not bad, maybe it's just a normal polymorphism in the genome. But because you haven't tested many and you can't really talk to other labs about it, you don't necessarily know. And Myriad has tested thousands and thousands of people, and so they have much more information on what's common, what people actually get breast cancer that have this mutation versus that one. So I think there'll be sort of a big opening up of some of this information, certainly on those 2 genes and on others as well.
Great. Maybe they'll open their data.
I don't think probably won't.
Wishful thinking.
So another follow-up on that. can we still patent the techniques to sequence genes? Is that still an option?
Yeah, for sure you can, and I mean not the ones that have been around forever, obviously, but new ones. So if you came up with a new technique, and I think that's the thing that most people are pushing for is don't patent the sequence, and don't patent maybe the knowledge about the sequence, but if you want to patent a technique, then there's no reason why you can't do that. And so there's new sequencing techniques that are always coming out, and they're, I'm sure, heavily protected by patents.
Okay, and then a third question from the community. Skylar Hayes asks, what does this mean about gene splicing? Can a company take, say, a similar primate gene, splice it with a human gene, and then patent it?
Well, they probably could. I mean, so a primate gene, maybe it's identical to a human gene, probably not. I mean, I think it really goes back to how much have you changed? So we were talking about the cDNAs and how much change do you need for it to be patentable? So the Supreme Court said, cDNA has got some modifications to it, so you can patent that because you've modified it, you've come up with this creation yourself, and so you can protect that with a patent. But they didn't say how much of a change it is. So a straight cDNA, has no introns, so you sort of change that. But I would argue that you really haven't. So if you change one nucleotide, is that patentable? I don't know. And so there's this sort of this range, and as Nick said, there's a good Nature article on this. They call this sort of this rheostat of changes, and how much change is enough to warrant a patent, and there's some threshold, and no one knows what that threshold is, I suspect. And so hopefully there'll be some sort of functional, like what does that one change? So if you change one nucleotide, but it makes a completely different, unexpected thing, then maybe. But if you just kind of take one bit of an ape gene and stick it onto another bit of a human gene, I think that probably there's going to be difficulty in patenting things like that.
So a couple more questions from the community. We have from Sancho Macan. We have, is isolation in fact necessary to perform genetic testing? And if so, do you expect that isolation would always be necessary to perform genetic testing?
Yeah, so generally it is. So you've got the DNA in your cells and all these chromosomes, but it's difficult to do anything with just a small number of molecules. So the isolation that generally is done in the lab is to do some sort of PCR to make many millions of copies, and then you can look at all those. PCR.
Define this acronym.
Oh, polymerase chain reaction. Yeah.
Okay.
So,
thank you.
It's a way. It's this technology for taking one or very few numbers of DNA, and it's an enzymatic reaction that makes millions of copies. It's like a DNA photocopier, and it's been used in the lab for, sort of 30 years, almost 30 years. And so it's very common, and so generally that's done. So in terms of isolation, you don't want to work with an entire chromosome that's millions and millions of bases long. You want to work with a small segment. So you PCR amplify to make copies, and you put things on the end so you can do whatever manipulation you want. There's some technologies. So there's these nanopore sequencers that everyone, they're on the cusp. They've been on the cusp for 2 years, and I'm so excited about these things. But you can sequence DNA. Basically, there's a very microscopic hole, and as the DNA kind of threads its way through, like a snake through a hole, it's able to read the bases of the sequence. And so that's not really isolation. You break open the cell, and the DNA gets fed through somehow, and you read it. So there's probably going to be technologies that don't involve all this amplification and segmenting the genome, because those are the lab processes that take a long time. So soon, I guess.
Soon.
Yeah. The follow-up to that, and interestingly enough, I think this gets toward the concept of the sequence being a molecule versus the sequence being the information, and that's sort of, I think, where the biochemists and the geneticists are really trying to wrap our minds around some of these things. But the follow-up to that is there any existing or imagined use of a genetic sequence that would, that doesn't require isolation? So I mean, I guess this is getting at, is having the sequence enough, or do you have to actually have the physical molecule for anything that would be of use?
That's a good question. I mean, in a lot of cases, the sequence having the sequence is enough. Like outside of a lab, like so if you're doing a research lab, you really need those bits because you're doing research on something and you need to build this or build that or some recombinant thing. If you had if someone's complete genomic sequence and you really, even if you needed to build some recombinant DNA, you could do that synthetically. You can make short segments of DNA synthetically. So I guess it's possible having sort of, a complete knowledge of someone's sequence without actually sequencing them is still very, very useful. Certainly for like genetics and genetic diagnostics, that's what we want anyway. If you could just wave a magic wand and know the sequence without all this messy, wet bench stuff, then that would be the best anyway.
Okay, very good. Well, we have about 4 minutes left. There's a couple additional questions kind of on the event page, including Robert Jacobson asked here, from what you know, do you feel that the justices adequately understood the science and technology involved in this case to fairly adjudicate the case?
I didn't really get the sense that they did understand it. That was my first I read it and I thought, wow, do you not have people that, I'm sure like a scientist at NIH would gladly come down and spend like 2 days just educating them, sort of nonpartisan, just this is the science. There was a few mistakes. Sort of the composite DNA was a bit of everyone kind of picked up on that right away because it's just the wrong. It's not the name that's used. Like there's nothing wrong with it. It's actually not a bad description, but it's just not the right word. There was a couple of comments about pseudogenes that weren't quite right. They said the lab technician removes the introns, and that's not the case. I think that's a subtle point, but I think that's an important point. So they got it. They didn't maybe get it as much as I would have hoped.
Oh, here's an interesting question. We have a few minutes left from John Malthus. A question for anyone. Is there any way of knowing whether a particular manipulation done in a lab somewhere would have been possible under normal evolutionary processes?
It's biology, so I never say never or always. So probably, if you look at cancer, the chromosomes do incredibly strange things, really weird. So it's entirely possible that something that you thought up has been somewhere before in nature. In the ruling, they kind of actually said, we're accepting the really rare situations where, pseudogenes do this or that. So they tried to acknowledge that there's some really rare cases where DNA does funny stuff. But you can never say never. It probably is somewhere.
Okay. Well, I want to end with a question for you, Ian, just about why are you passionate about genetics? What about this field got you interested in this field of science to begin with, and do you have any words for possibly young people who might be interested in pursuing a career in genetics or genomics?
It's a lot of fun. I don't know. I really like DNA. I kind of liked it in high school and university. I started out in chemistry actually, and I was like, no, I like biology better. I just have a passion for biology. Genomics is this great sort of cross-disciplinary thing. If you're really good at computer science or statistics, you can actually, but you're interested in DNA, you can actually find, great work and great positions doing bioinformatics. There's a lot of, there's wet bench stuff. There's a lot of, it overlaps with medicine as well. And I think if you're interested in medicine, then you really have to be interested in genetics as well. And I really just enjoy working on cancer. So, for personal reasons and just for professional reasons, it's really satisfying to try and come up with these genetic answers that'll help people.
Excellent. Well, that's a great answer. This has been very fun and very informative. I have learned a lot in this hangout and preparing for this hangout. Thank you so much, Ian, for joining us, and
Thanks.
Jason and Nick for co-hosting our very first Science on Google Plus live conversation. Actually, our next one is coming up with Joe Hansen of It's Okay to Be Smart, and that's taking place, when is it? That's the 23rd of July, so be sure to RSVP for that. It should be very fun. We'll be talking about gifts and science and how to share it with the world and get people excited about the awesomeness that the wonderful world of molecules actually is. Joe is actually also a biologist. So I guess with that, we'll sign off And this video will be archived forever on YouTube. Let us know what you thought of the Hangout in the comments, and maybe Ian will even answer some additional questions if he has some time.
Yeah, for sure.
All right. So thank you, guys. Goodbye.
Thanks.
Thanks.
Thanks.